The seed definition is important. So once again, I'll remind you. You remember once when we were discussing about the flower and the ovules, I had explained to you there that ovule, the number of the ovules in the flower decides the number of the seeds in the fruit. Yes, decides the number of the seeds in the fruit. The example was given was papaya and watermelon. You all are aware about it. Yes, many numbers, multiple number of seeds are present in those fruits. Yes, multiple number of seeds are present in those fruits. That means what? There are many large number of ovules in it. How many? Many number of ovules in that flower. Now here, what you have to learn is that ovule, the seed is nothing but a fertilized ovule, or it is a seed is a post-fertilization product of ovule. Means it develops from ovule. See what you have to remember here. Ovule develops from, sorry, seed develops from ovule. Seed develops from ovule. It is a fertilized product. Yes, seed is a fertilized product of ovule. Seed is a fertilized product of ovule. So first you need to remember the definition. Seed is a fertilization product of ovule, or seed is a fertilized ovule. The definition is clear? Yes. Uh, is it clear about the definition? So, seed is a post-fertilization product, or seed is a fertilized ovule. Okay, next coming to seed consists of. Yes, we studied about, when we were studying about the embryo, we studied about the embryonal axis. Yes, the embryonal axis consists of the plumule, a radicle, a root cap, coleoptile, coleorhiza, and in monocot it is scutellum. Yes or no? The cotyledons are called as scutellum. The cotyledons are nothing but the covering. Okay, cotyledons are nothing but the covering of the embryo or covering of the seed. If it is one covering, then it is called as monocot. If there are two coverings, it is called as dicot. Yes, if there are two coverings, then it is called as dicot. And if it is called, if it has got only one covering, it is called as monocot. Mono one, di two. Okay, so here the seed consists of cotyledons, that is the coverings. Okay, cotyledon it can be for monocot, and cotyledons it can be for dicot. Then coming to embryonal axis. In embryonal axis, what and all is there? There consists of scutellum, coleoptile, plumule, which is the shoot part, radicle, which is the root part. Yes or no? The radicle, which is the root part. Now, seed coat is double layer. How many layer? Double layer means two layer formed from the integuments. You remember students? Integuments was present. This is the megasporangium and these are the integuments presented. Yes or no? The two coverings for the ovule. The two coverings for the ovule. This is the outer integument. This is the inner integument. Yes or no? Outer integument and inner integument. Yes. So this outer integument forms into outer coat known as testa. What it is known as? Testa. It is known as what? Testa. The outer integument forms into outer coat which is known as testa. The inner integument which is the inner coat is known as tegmen. What it is known as? Tegmen. Is it clear? The inner integument is known as tegmen and the outer coat or the outer integument is known as testa. Okay, it is known as testa. So it is a double layer. So it is a double layer formed by integument. The seed coat is double layered formed by integument. Okay, can you see your students seed coat and the fruit wall? Yes. Now, whenever I ask you, how many seed coats are there? There are how many seed coats? Two seed coats. The one is the outer seed coat, which is testa, and another one is inner seed coat, which is tegment. Yes or no? Two seed coats, outer seed coat and inner seed coat. Okay. Yes. Now coming to micropyle. Can anyone tell me what is a micropyle in their own words? So, yes, where there is a small opening called as micropyle. Okay, there is a small opening called as micropyle, and this micropyle helps in the movement of the pollen tube. Helps in the movement of the pollen tube. So this micropyle or the small opening, this micropyle or the small opening also remains in the seed coat. Also remains in the seed coat, and it helps in the entry of the water and oxygen inside the seed. It helps in the movement of the oxygen and water inside the seed. So, see, this is a seed. Okay. So there will be one small opening here. Where it will be? Here, a small opening. It is known as micropyle. Now, what does this micropyle do? This micropyle helps in the movement, that helps in the movement of the water and oxygen inside and outside the seed. Okay, inside and outside that seed. It helps in the movement of it. So micropyle is nothing but a small opening on the seed coat. A small opening on the seed coat which helps in the movement of the water and oxygen. Okay, so it is micropyle, and micropyle is also present in the ovule. You remember, micropyle is also present in the ovule, the megasporangium. The ovule helps where it helps in the pollen tube to enter inside the embryo sac, where it helps the pollen tube to enter inside the embryo sac. So, because it is present in the ovule, it is also present in the seed. Yes, it also present in the seed. Now, coming to perisperm. Now, this is important, students. The question about the perisperm can be asked in your exams, for your competitive exams as well. So, what is this perisperm now? Perisperm is nothing but the remaining of the nucellus part. Now, you will ask me, what is nucellus, ma'am? See, beside this embryo sac, there is something tissues. Yes or no? The chalazal part and all, there are tissues right here. These tissues are not, this tissue is nothing but called as nucellus. What it is called as? Nucellus. Now, if the surrounding tissue beside the embryo sac, it remains in the seed also. If it remains in the seed also, then it is called as perisperm. What it is called as? Perisperm. Remnants of nucellus that is persistent. Yes, there is a remaining of this perisperm. And the example for it is black pepper. Black pepper, can you see here? These are the black peppers. Okay. So now, for you, this question can be asked in this way. Okay. Which among these seeds have perisperm in it? Yes. Now the question can be asked. Which among these seeds have perisperm in it? In that one option will be paddy seed. One option will be black pepper. One option will be something of wheat, or another option will be something of any other. Okay, then the answer for this is black pepper. Black pepper has the remnants of nucellus. Black pepper has the remnants of nucellus. So perisperm is nothing but remnants of nucellus, and the example is black pepper. Example is what? Black pepper. Okay, so this is about the perisperm. Next, coming to dormancy. Now, what do you mean by dormancy? Dormancy is nothing but state of inactivity. Means inactivity means no activity is taking place. There is no activity at all. Now, what happens here? For example, think Aditya has got one seed. Yes, Aditya has got one seed and he is trying to germinate it. Yes, he is trying to germinate it. But there is no proper water and there is no proper sunlight there. Neither there is no proper, the soil doesn't have any kind of nutrients. Okay, no, doesn't have any kind of nutrients. Now, what happens here? Even though he is trying to germinate, he is trying to germinate that seed has adapted itself in that environment, in that surrounding. That means what? it has undergone state of inactivity. Inactivity means there is no activity at all. Yes, there is no activity at all. Means the seed is not doing anything. It is not germinating. It is not allowing the seedling to grow. Yes, it is not allowing the seedling to grow. There is nothing happening there. So that means it has undergone the state of inactivity. And why it has undergone the state of inactivity? Because there is no proper sunlight, water for the seed to grow. Yes, and that state is known as dormancy. That state is known as dormancy. The state of inactivity of the seed is known as dormancy. And now seed and embryo. This is just an example, okay? Seed and embryo is an asked field. Now what you will do? One definition of seed you will write. This definition. One definition of embryo you will write. This is for 2 marks. Now next you need to give another one definition or another something you need to give the differentiation. So here you can give the example. Here you can give the example. Yes. So example helps you, right? Yes. Example will help you here. So why I am stressing you to even learn the definition and also the examples. Yes. That rice will have one red color layer on it. Okay, a red color layer. And that red color layer is nothing but called as alluron layer. Alluron layer. Can you see here? This is something new, no? Limule, scutellum, coleoptile, endosperm, you have already studied it. Yes or no? Coleorhiza in the embryo. Whereas the alluron layer is something new. Now, what is this alluron layer? Now, in this rice, I have told you one red color coat will be there. A red coat. Yes or no? So, now, if you get this golden rice also, in the from the shop you get one golden rice. Yes, Sona Masuri rice. You get that golden rice. Now, people usually say that it is a good rice. Yes or no? People say that it's a healthy rice. You need to eat it. Now, why it is being told that it's a healthy rice is because it contains this alluron layer, that golden layer which is present there is nothing but the alluron layer. Okay, but that alluron layer there is added. Okay, that golden rice, golden color polish is given to it. A gene known as alluron gene is inserted. But when we see this in the normal rice which we grow in the fields, with the red coat which we get, that is also alluron. So you do not have to, you do not have to remove that polish, or you do not have to separate that water. Actually it has to be taken because it is rich in vitamin A. It is rich in vitamin A. Okay, so that layer is nothing but the alluron layer will be present here. Okay, the alluron layer will be present here, and this alluron layer is nothing but a vitamin rich layer. Okay, that red coat which I am telling in the this boiled rice also, this is also very nutritional. It contains vitamin A, whereas in the golden rice also it contains the gene alluron in it. So it is also rich in vitamin. It is also rich in vitamin. So we say that the golden rice is a healthy rice. It's because of the presence of the alluron gene in it. Alluron gene in it. Is clear now? Yes. And also that rice which I am telling you, that the boiled rice which I am telling you now, that which has that red color coat, that coat is nothing but called as alluron layer. It is called as what? Alluron layer. So different stages of embryo development in dicots. Students, you need to remember first step, embryogenesis process starts with zygote. Where it starts with? It starts with zygote. Okay, and how the zygote is formed? The zygote forms from the fertilization process, that is the male gamete and the female gamete, male gamete and the female gamete fertilize to give a zygote. Yes, this is nothing but the fertilization process to give the zygote. Now, this zygote later develops into embryo. Yes, develops into embryo, and that process, the process of the formation of embryo, the process of the formation of embryo from the zygote is known as embryogenesis. What it is known as? Embryogenesis. Yes, this process is known as embryogenesis. Now, what happens here? This process is known as embryogenesis. The process of development of an embryo from the zygote is known as embryogenesis. Now, remember, first learn the diagram. Once you learn the diagram, it becomes easier for you to understand the steps. Okay, so here, this is a zygote. Now, the zygote undergoes mitosis. What it undergoes? Mitosis. And mitosis is nothing but equational division. Yes. Zygote undergoes mitosis forming two cells. One is the apical cell and another one is basal cell or the terminal cell or the basal cell. It forms two cells. Yes, it forms two cells. One is known as the basal cell and another one is apical or the terminal cell. Understood? The zygote undergoes mitosis and it divides to form two cells. Now, what happens here? The apical cell forms into two-celled embryo. It further undergoes division. It further undergoes these two cells further undergoes division. The apical cell and the basal cell further undergoes division to form two-celled embryo, and the 6 to 10 cell structure called as suspensor. 6 to 10 celled structure called as suspensor. Yes, the basal cell forms into suspensor, the apical cell forms into two-celled embryo. Is it clear till here? The terminal cell or the apical cell forms into two-cell embryo and the basal cell forms into the 6 to 10 cell suspensor. Later what happens? Here the embryo, 4-cell embryo, again further division takes place, 4-celled embryo is formed and the suspensor, 6 to 10 cell structure known as suspensor. Later, further it divides to form globular embryo. Here the embryo forms into round structure along with the suspensor. Along with the suspensor. And this we call it as globular structure. Later, from the globular structure, it further divides to form a heart-shaped embryo. Yes, a heart-shaped embryo here. Can you see here? It is a kind of a heart shape, right? And this is nothing but a suspensor. Yes, this we call it as heart-shaped embryo. See, it started from the zygote. The zygote divided into two cells, apical cell and basal cell by the division of mitosis. After that, further division took place to form two-cell embryo and the suspensor. Later, it further divided to form the embryo and the suspensor. Later, further it divided to form a globular stage, that is the globular embryo along with the suspensor, and from the globular stage, it formed into a heart-shaped embryo and the suspensor. Later, after formation into this heart shape, it further divides to form a mature embryo. It further divides to form a mature embryo. And because it is a dicot, there are two cotyledons. There is a shoot part. There is a shoot part. This is the suspensor part, and this is the radical. That is the root meristem. The shoot meristem is called as a lemur. The shoot meristem is called as a lemur. From the heart-shaped It further divides to form a mature embryo. It further divides to form a mature embryo. And this entire process is known as embryogenesis. The entire process is known as embryogenesis.